Okay. So today I'm going to good morning. Today I'm going to present on fuel for exercise. So bioenergetics and muscle metabolism. So what is meant by energy intake and energy expenditure? How much energy being produced while we are walking, running, and hiking lah. So how are you getting this energy from? So how you're getting all this energy from is from these three basic fuels, so which is known as substrates as well. So there is carbohydrates, proteins, and fat. So it will be broken down to release the stored energy lah. Okay. So what is meant by bioenergetics? So it's a chemical pathway which can convert the substrate into energy. So that is meant by bioenergetics. So metabolism is chemical reaction that happens in the body. So what is ATP? So ATP is the one I said, energy in the food molecular bonds, which is chemically released within our cells lah, and stored in the form of ATP. So energy is ATP. So just remember that lah. So this is a small diagram. So which is that when you take food in, means there's a food intake. So it will be divided into three: carbohydrates, proteins, and fats. So what will happen when it goes inside our body, in our body? So it will broken down to glucose lah. So from glucose, it will further broken down to glucose pool. So this glucose pool is what happens whereby the energy being produced in this glucose pool lah. So if let's say there's too much of glucose, then it will be stored in liver and muscle as a glycogen. So that is the process we call as glycogenesis. So in the process of glycogenolysis, the glycogen will be broken down to glucose. So whereby the energy to produce energy lah. So basically, so one is to produce and one is to keep. So lysis means we actually to produce energy lah. Okay, and genesis is to keep the energy. So this is how I remember lah. So then it goes with this free fatty acids, the fats actually. So fats is divided into it will broken down to free fatty acids and glycerol acid. Okay, free fatty acid with glycerol. So the primary energy source usually free fatty acid lah. So in this one, so energy will be produced by the process of will be produced in the FFA pool lah. So from the fat store, okay, let's say if there's excess of fat, so it will be stored in adipocyte tissue or cells lah. So if let's say too much of fat, you want to burn it, so we call if you want to break down, so the process is lipolysis lah. If let's say it is you want to keep the fat, so means it's lipogenesis lah. So the same thing goes with the protein as well. Okay, so this pools is basically happens in all this metabolism happens in the blood actually lah. Okay, the amino acid, glucose, and free fatty acid pool. Okay, so how we decide on the rate of energy be produced lah. So the first thing, availability of the primary substrate. So we must see whether this carbohydrate is more than this protein or protein is more than fat lah. So then we decide whether we can take the energy from carbohydrate or fat or protein. So the next one is the other factors by enzyme activity lah. So influence of substrate availability on rate of metabolism is what we call as mass action effect. So if let's say carbohydrate, we are using carbohydrate in the rate of metabolism, so that is we call as mass action effect lah. So this is a diagram. So whereby in order to start a chemical reaction in our body, so activation energy is needed lah. So how much is depending on the enzyme actually. So whether you need enzyme or don't need enzyme. So if you need enzyme, so the activation energy will be lower than the one without enzyme. So that is how happens lah. So rate limiting enzyme. So what is meant by rate limiting enzyme? So it's an enzyme that is of particular importance in controlling the rate. So it's all throughout all the pathways that I'm going to explain later. So it will decide whether which will be, how much energy will be produced lah. So this rate limiting enzyme lah. So their activity usually determined by accumulation of substances further down the pathway that decreases the enzyme activity through the negative feedback lah. So if you see, like I explained this now, if activation energy is needed in order for the pathway to occur, so in the end, so the byproduct, so if you see ATP will be released, okay, and if let's say you want to use it as energy, ADP, and it will break down to ADP and phosphate lah, inorganic phosphate lah. So yeah, so usually the rate limiting enzyme will be the first enzyme to be in the pathway lah. So followed by others. So the next one is storing energy, high energy phosphate. So this one is adenine and this is ribose. So why I explain this is because want to know how this ATP goes to ADP lah. So this one there has a high energy bond, actually, the phosphate. So if you see, this adenine has nitrogen base and this ribose has carbon base. So and this is the phosphates lah. So this is what we call as adenosine. Okay, this is adenosine and this is the phosphate, 3-phosphate, so triphosphate. So what happened is that add on the water, so in the process of hydrolysis with the help of enzyme of ATPase, so it break down to adenosine with 2 phosphates, and then we release another phosphate and then with free energy lah. So this is structure of ATP lah, like what I said just now. Okay, so the next one is basic energy system. So I'll just go roughly on this generation of ATP, how ATP is being produced. So the first one is by the ATP PCR system. The second one is glycolytic system, so which is we call as glycolysis. So this is without oxygen, so anaerobic. So the third one is oxidative system, so which is we call as oxidative phosphorylation lah, with oxygen. So if it's with oxygen, then it's aerobic lah. So ATP PCR is classified as substrate level metabolism. So what happened is this phosphocreatinine will release this phosphate, so in the help of this enzyme of creatinine. So what happened is that this phosphate and ADP will combine and form this ATP lah. So that is how it happens. So if it's ATP is in a stored energy, ADP is energy that we gonna used it lah. So that is how we remember lah. So in this ATP PCR, ATP can be recreated by binding the inorganic phosphate. So that is about ATP PCR system. So intense exercise is initiated. When intense exercise is initiated, ATP in muscle cells will be broken down. So it will yield on the ADP and inorganic phosphate. So it causes increased ADP concentration, and hence it causes increased creatinine kinase activity. So when that happens, this PCR will be catabolized to form this additional ATP. So as the exercise progresses and oxidation, ATP is being generated by other two systems. So and the creatinine kinase activity will be inhibited lah. So this is a graph. So in the x-axis, if you see, is the time, and the y-axis is the percentage of resting volume lah. So changes in type 2 fast twitch skeletal muscle adenosine triphosphate, and also the phosphocreatinine PCR during the 14 second of maximum muscular effort sprinting. So the black one is the ATP. So, and the red one is PCR. So what happens is that as ATP being used, so to avoid the ATP to be in the decreasing value, so the PCR will back up. So means PCR will support the ATP, so it will give more energy to ATP lah. So at the end point there, if you see the 14 second there, these two will be having an exhaustion lah. So they will have exhaustion. So that's what I meant actually, is like a combination of this ATP and PCR stores can sustain the muscle energy need for 3 to 15 seconds during an all-out sprint. So beyond this time, so muscle must rely on other process for ATP formation lah. So it means only for about 3 to 15 seconds only. So after that, it will go to other processes lah to rely on to generate energy lah. So the other system that I would explain is about glycolytic system. So it is breakdown of glucose through a pathway that involves a sequence of glycolytic enzymes. So basically it's a breaking down of glucose lah. So predominate usually early minutes of the high intensity exercise. So major limitation of anaerobic glycolysis accumulation of lactic acid in muscles and body fluids. So how it happens? So when glycolysis produces pyruvic acid, so without pyruvic acid converted directly into the lactic acid lah. So for this glycolysis pathway, usually the rate limiting enzyme is phosphofructokinase or PFK. So what it does actually, it catalyzes early step in pathway. So from the conversion of the fructose 6-phosphate to fructose 1,6-phosphate lah. So when there's increased ADP and inorganic phosphate, there will be increased PFK activity, therefore it speed up the glycolysis lah. So if let's say meanwhile increased ATP will cause decrease in PFK and slows down the glycolysis. So if you see the process actually from glycogen, how it goes to glucose and how it produces this pyruvate, and in the end it will produce this lactic acid lah. So this is without the oxygen, so we call it as anaerobic glycolysis. So for all this process, right, so if let's say the net gain is starting, if the process is starting from glucose, so the net gain ATP you can get only 2. If let's say from glycogen, the ATP you can get is 3. So okay, so if you see from all these glucose, glycogen, glucose 1-phosphate, glucose 6-phosphate, glucose, and then it goes down, breaks down to fructose 6-phosphate, and from fructose 6-phosphate breaks down to fructose 1,6-diphosphate. So meanwhile, you see the ATP is being broken down to ADP to generate the energy lah. So all this while, all the hydrogen, it being, okay, if you see here, there's NAD. So NAD is a coenzyme lah. NAD will be in a reduced form. So we release this hydrogen. So what will happen is this, the hydrogen will be transported to the electron transport chain. Why is it? Because they don't want too much of hydrogen ion in the pathway. So it causes more acidification lah. So that's why they transport the electron transport chain. So I will explain how later on. So okay, so this one is okay. The next one I'll talk about oxidative system lah. So oxidative system is a final system. So most complex one is very slow to turn on. So it has a larger energy producing capacity. So hence aerobic metabolism for endurance activities lah. So that's meant by the cellular respiration is process by which body breaks down substrates with aid of oxygen lah. So anaerobic usually happens in cytoplasm. Aerobic happens in mitochondria. So oxidative of carbohydrate. So what is oxidative? There's 3 pathways. The first one is glycolysis, Krebs cycle, and electron transport chain. So in this glycolysis, if anaerobic, this pyruvic acid will be converted to lactic acid lah. So whereby I just now told you the 3 net mole of ATP will be produced if glycogen, or 2 net of mole of ATP for the glucose lah. So for aerobic, usually the pyruvic acid will be going to the acetyl-CoA lah. So this is with the aid of oxygen. So this Krebs cycle is a citric acid cycle, or we call it as tricyclic acid cycle. So it's a complex series of the chemical reaction that permit the complete oxidation of acetyl-CoA. So whereby the pyruvate from glucose to pyruvate and go to the acetyl-CoA lah. So this glucose to pyruvate, right? So it happens in cytoplasm lah, cytosol. So what happens is this, glucose has 6 carbon sugars. The pyruvate will further divide into 2 pyruvate lah. So that means it will have 2 pyruvates with 3 carbon in each. So it will divide. So how is it with the energy that they have lah, ATP? And then at this same time, so the coenzyme NAD will be reduced and they will release this hydrogen ion. So the same thing from pyruvate goes to acetyl-CoA. Okay, the same thing also lah. So it releases CO2 as well. So this thing happens in mitochondria lah. So why is it we call as a Krebs cycle? If you see, right, it's a cycle, right? So the initial product will be the same as the end product. So that's why it's called as cycle and it's not called as pathway lah. So if it's called pathway, means be followed by. But if you call it Krebs cycles, mean you are getting the same end product. So which is the acetyl-CoA. So what happened is that, so all this hydrogen ion will be released. Okay, so it will be going to this mitochondria matrix. So through that, okay, they will they will change to electron. So the electron will pass from one complex to another complex. So that's why it's called as electron transport chain. So what happened is that there will be oxygen there. So the hydrogen will mix with this oxygen and will produce this water. So why is this? Because to avoid the acidification of the cell lah, basically. So basically this Krebs cycle is being coupled with this electron transport chain. So all this process where they yields about 28 ATP lah. Okay. So this is what meant by the Krebs cycle roughly. So okay, so this is the more like a detailed version, you see. So the Krebs cycle. So from pyruvate, how it goes to acetyl-CoA. So the rate limiting enzyme, so you just need to know about the rate limiting enzyme is isocitrate dehydrogenase, which is the first out of all the other enzymes lah. So it decides how many energy being produced lah. So okay, if you see in this case, there will be a GDP. So what is GDP and GTP? It's almost same like ATP and ADP lah. Okay, so then okay, end of this Krebs cycle, 2 additional ATP form directly. So carbohydrate will be broken down to carbon dioxide and H2O. So this the end product will be carbon dioxide and also H2O. So the rate limiting enzyme, like I said, is a isocitrate dehydrogenase. So it will be inhibited by ATP and activated by ADP and inorganic phosphate. Okay, so this is what I explained just now, electron transport chain. Okay, so energy yield from the oxidation of this carbohydrate. So how much of energy from this oxidation, how much energy you are getting, the energy is like total is about 32 ATP. Okay, if this happens through the glucose, if let's say from glycogen, it's 33. So there's another, the next one I'll talk is oxidation of fat lah. So to be used as energy actually, so triglyceride will break down to glycerol, 1 glycerol and 3 free fatty acids. So with the help of lipase, the enzyme of lipase, and we call the process as lipolysis lah. So what happens is this FFA is a primary energy source for fat metabolism. So it enters blood and then it enters the muscle via the simple diffusion or facilitated diffusion now. So rate of entry into the muscle fibers is depending on the concentration of gradient now. So if let's say increase concentration of free fatty acid in blood, so there will be increased rate of transport into the muscle fibers. Okay, that is about the oxidation lah. So the next thing I'm going to talk is about beta oxidation. So in order to use the free fatty acid as the energy source, so we must be converted to acetyl-CoA in mitochondria. So that is what we call as beta oxidation. So whereby there's a series of steps in which the 2 carbon acyl units will be chopped off the carbon chain of free fatty acids lah. So I'll give you an example lah. So if let's say free fatty acid has about 14 to 24 carbons. So via this beta oxidation, so they will yield about 8 molecules of acetyl-CoA, half of it lah. Okay, so what happened is this, the acyl unit becomes acetyl-CoA, which then will enter the Krebs cycle for ATP formation. So this is what happens. So maximum rate of ATP production from lipid oxidation is too low to match with the utilization of ATP during high intensity exercise. So that's why when reduced athletes face lah, so when the carbohydrate stores depleted and they use fat as the predominant fuel lah. So the next one is Krebs cycle and electron transport chain for the fat actually. So after the beta oxidation, fat metabolism follows similar pathway as the oxidative carbohydrate metabolism now. So acetyl-CoA will be formed by the beta oxidation, so it will enter the Krebs cycle. So Krebs cycle will generate the hydrogen and transport the electron transport chain along the hydrogen with the hydrogen generated. So during the beta oxidation to undergo the oxidative phosphorylation lah. So oxidative phosphorylation is ADP adding on the phosphate. So that is what we call as phosphorylation lah. So byproducts of free fatty acid oxidation, there will be an ATP, H2O and also carbon dioxide lah. So complete combustion of this free fatty acid molecule requires more oxygen because free fatty acid contains more carbon molecule compared to glucose. So free fatty acid has more carbon compared to carbohydrate lah, that's why. So more carbon molecules, more acetyl-CoA, more electrons sent to electron transport chain, so more energy than the glucose metabolism lah. So this is just an example lah, palmitic acid. So it has about 16 carbon free fatty acid. So oxidation, Krebs cycle, electron transport chain will occur. So how much of energy we getting from here is because roughly about 129 molecules of ATP. So which is from this one molecule of palmitic acid. So the next one I'm going to talk about the oxidation of protein lah. So protein is being used for energy and use energy for under some circumstances lah. So when there is amino acid being catabolized, some of the released nitrogen be used to form a new amino acid. So they cannot be oxidized. The nitrogen cannot be oxidized. So they only can change to amino acid or produce a new amino acid. So what will happen is that it will convert to urea and then excreted via urine. So this conversion will require use of ATP. So some energy will be spent in this process lah. So to assess on this rate of protein metabolism, amount of nitrogen being eliminated from the body must be determined. Okay, so this is roughly about the summary of the substrate metabolism lah. So what really happens? So basically glucose will be absorbed in the intestine. So then glucose in the blood vessel, and then this is energy supply and energy demand. So what will happen is that the glycogen will break down to pyruvate with the process of glycolysis. Okay, if you do the exercise lah. If at rest means it becomes glycogen. So then it goes this oxidative phosphorylation lah, which is the scrap cycle. Okay, to get all this energy. So what they're going to do with this energy? So basically ATP then we change to ADP to an inorganic phosphate in order to get a contraction. So with the help of the myosin ATPase. So the other one is ATP with the creatinine lah. So if let's say ADP, it will convert to ADP to produce a contraction lah. So the same thing goes with this one actually for the liver. Glycogen will turn to glucose, and glucose will go to the same process lah. So then the lipid will be stored in adipose tissue, will break down to glycerol and free fatty acid. So in blood vessel it will be free fatty acid and then convert to acetyl-CoA. So from acetyl-CoA goes to the Krebs cycle. So this is where the energy being produced, ATP. So from ATP you convert to ADP for the energy. So the same thing with this exchange of gases lah, oxygen and carbon dioxide. So oxygen will go to this aerobic, you need oxygen, right? So from the lung will go to the Krebs cycle lah. So basically, so the next one I'm going to talk about interaction among the energy system. Okay, so 10 second 100 meter sprint. So ATP PCR is the predominant lah. So why is it? Because it provide energy at very fast rate. So other system provide but very small portion lah. Then the next one is 30 minute 10,000 meter or otherwise 10,936 yard. So in this process, oxidative system will be the predominant. Why? Because provide energy at slower rate and can give a lot of energy. I mean unlimited of amount of energy lah. ATP, PCR and anaerobic glycolysis will contribute some of the energy. So oxidative capacity of a muscle lah. So is a measure of its maximal capacity to use its oxygen. So how to measure? Small amount of muscle tissue will be tested in the lab. So they do a biopsy actually lah, to determine the capacity to consume oxygen when chemically stimulated to generate the ATP. So enzyme activity wise, measuring the enzyme. So reflecting aerobic capacity of fibers lah. So most frequently measured enzyme is succinate dehydrogenase and citrate synthase with other mitochondrial enzymes in Krebs cycle lah. So fiber type composition and the endurance training. So as previous muscle fiber type composition determines the oxidative capacity lah. So in previous presentation I got mentioned about this type 1, which is slow twitch, and type 2 is the fast twitch. So this type 1 has a greater capacity for the aerobic capacity activity lah, and it has more mitochondria and higher concentration of oxidative enzyme lah. So for type 1 usually elite distance runner lah. For type 2 usually better suited for glycolytic enzyme production. So example, this is for endurance training lah. So oxygen needs, oxidative capacity of muscle is determined by these three things. So number of mitochondria, amount of oxidative enzyme present, and oxygen supply. So when the exercise intensity increases, the energy demand will be increased as well. So the rate of oxidative ATP production will be increased as well. So in order to meet this muscle need for oxygen, okay, the rate and the depth of respiration will be increased. So the next one, this is on the lungs. The second one, heartbeat will be faster and more forcefully lah, the pump. So to pump more oxygenated blood to the muscles. The third one is the blood vessel, dilatation of the arterioles to facilitate delivery of arterial blood to the muscle. So amount of oxygen entering the blood as it passes the lungs directly proportional to the amount used by the tissue for oxidative metabolism. That's all.